Hey there, awesome disease and public health students. I'm gonna just walk you through how to make the drawing about non-specific defenses, specifically how natural killer cells and complement proteins can lyse pathogen cells or abnormal cells. First, you're gonna need a sheet of scratch paper. I'm gonna use an old chemistry worksheet for acid-base stuff. Cross that out and use the blank side of it. So you just need a blank piece of paper. If you don't have a blank piece of paper, you can just rip a piece of lined paper out of your notebook or something like that to use. I'm going to use a Sharpie just to be a little clearer here. At the top of the page, you're going to write immune system. And specifically, we're talking about non-specific defense. This is just going to be one example of that. Now, on the left side, you're going to write natural killer, or what we call NK cells. And on the right side, you're going to write complement proteins. We're going to look at how both of these systems work. First thing you're going to do is draw an arrow going straight down from each of these to show the tool they use. Natural killer cells use proteins called perforins. Draw a box around that. Complement proteins form what is called a membrane attack complex. Membrane attack complex, or MAC for short. Again, draw a box around that too. Both of these work in the same way, and so what I'm going to do is just draw an arrow coming out from that one and an arrow coming out from this one, have them join in the middle here, because they're both going to operate the same way. Now, to demonstrate how these operate, I'm just going to go a little bit further down here. Let's actually do it. So both of these proteins punch holes in the membrane. And so what you can do is just put down a sheet of paper and, put your paper on top of a towel or a piece of cloth or something like that, and then literally just use a pencil or pen to punch a series of holes in the paper. Now in reality, what's actually happening is proteins are being embedded in the cell membrane, and that's what forms a hole in the cell membrane. But do it enough so that eventually you can just rip out that center of paper, and there you go. So let's now label what happened. You're going to go ahead and just draw sort of a series of boxes around your hole, like so. And those are either your, what are called the perforin proteins, or, that capital R, MAC proteins. That's what embeds and forms that little ring or hole. I draw just kind of a slightly curved line going across here, and let's label this as the surface of pathogen cell or abnormal cell. That's because this could also be like a cancerous cell that needs to be destroyed. Now we're just going to have 3 bullet points next to this hole to briefly summarize what happened in this process, okay? So the first step is that the proteins embed in the target cell membrane. The next step is that they self-assemble into a ring, forming a hole, which is what you did by punching those holes in and then ripping out that piece. And this is a problem for that target cell because now fluid rushes into the target cell causing it to burst, or what is more formally called lyse. And there you have it. That is how natural killer cells and complement proteins can destroy pathogen cells or abnormal cells. Submit a photo of your drawing as your contribution to the assignment. Hope this was helpful. Thanks.